Our next unit is going to involve something called protein synthesis. So as a reminder, synthesis is a vocabulary word that means either to make or to create. So the whole idea behind protein synthesis is to create proteins. So as you know, DNA is where all of our genetic information is stored. So DNA is the building blocks for all of the proteins that go about making up our body. The process and the kind of overall name of protein synthesis is called the central dogma of genetics. Unfortunately, it has nothing to do with dogs, but it is called the central dogma. Now again, think about it for a second. Where is our DNA? Right? Our DNA is in our nucleus. Unfortunately, the DNA does not have the ability to leave the nucleus. So all the instructions are in the nucleus, kind of like in the library, but you're unable to check out the book. So proteins are made in something called ribosomes. Now, as you recall, back in our lesson that involved organelles, we learned that ribosomes, the primary function of ribosomes is to make proteins. So, to overcome this hurdle, protein synthesis uses two different steps. Step one, where we go from DNA to something called RNA, ribonucleic acid, that's called transcription. Step two, when we go from RNA to protein, that is called translation. Now, there are three essential types of RNA. There are many other smaller types, but these are the three primary types we're going to hope that you are able to learn. The first type, mRNA. So your first letter in front of the RNA is going to be a dead giveaway for what type of RNA it's going to be. So mRNA, or messenger RNA, ultimately is just the carrier. So it copies the instructions from the DNA and takes that out of the nucleus into the ribosomes into the cytoplasm. So ultimately, this is kind of our UPS driver that's taking mail from the nucleus, delivering it to its home in the ribosome. tRNA, again, notice the T, or transfer RNA, carries something called amino acids to the ribosome and the mRNA. So if you look to the right, that is an example of amino acids. So you can see each amino acid ultimately is coded by three different letters. And our last one is called rRNA, so ribosomal RNA. This is what actually composes the ribosome. This is what puts the ribosome together and forms the ribosome. So again, the R is our dead giveaway there. So let's start with our first bit of information on transcription. So the whole purpose of transcription is to carry this code from the nucleus. So we're not making proteins yet. We're just trying to get the information from the nucleus out towards the ribosome. And again, just as a reminder, in no way does DNA ever leave the nucleus. If it was, that probably means the cell has popped or something went horribly wrong and is probably going to die. So this takes place in the nucleus. Your starting point is your DNA. And then that DNA ultimately interacts with our mRNA, and that mRNA takes the code from the nucleus. So here's another look at that. So recall, this is similar to our DNA replication process. So the gene gets unzipped, so very similar starting point. However, instead of DNA being formed, an RNA set of nucleotides ultimately pairs up with our DNA nucleotides that are open. So if you look below, the purple is going to be our DNA, and the red strand is going to be our RNA. So just remember, you can see on the DNA, the orange T's are thymines, but on the red, they are replaced with the purple uracil because RNA has uracil instead of thymine. So once the stretch of DNA is copied and the complementary mRNA strand is created, that mRNA is released. That DNA zips right back up and continues to function as it normally would, and the mRNA leaves the nucleus. So this is another way to look at it. So we start with our DNA. Ultimately, it splits. Our RNA comes in, matches up, and then our mRNA leaves and the DNA zips back up. So here's our DNA. Those are two parts of our DNA and our mRNA coming in. Then our DNA zips back together and our mRNA is given off. Now, let's see if you can figure this out. Try and determine the DNA sequence, and then try and determine whether the following sequence is appropriate. See if you can find any errors. So that is the gist of transcription.